Good morning. It's December 16th, and this is your daily brief in science. Here's everything you need to know. In 2023, the Mekong region of Southeast Asia has emerged as a focal point for biodiversity research, with the identification of over 230 new vertebrate and plant species. This extensive study, conducted by hundreds of experts over a year, includes a diverse array of findings: 173 vascular plants, 26 reptiles, 17 amphibians, 15 fish, and 3 mammals as reported by the World Wildlife Fund. The Mekong region, which spans Cambodia, Laos, Myanmar, Thailand, and Vietnam, is often referred to as a treasure trove of biodiversity. However, this rich ecological landscape is facing severe threats from habitat loss, illegal wildlife trade, climate change, pollution, and invasive species. Many of the newly discovered species are already at risk of extinction due to these human activities. WWF has called for enhanced protection measures from regional governments to safeguard these vulnerable species. Among the notable discoveries is a leafless orchid, Chiloschista kwangdangi, believed to be endangered due to habitat destruction and overexploitation. Other significant finds include a well-camouflaged lizard, Laodracon carsticola, and a hairy hedgehog named Hylomus macarong, after a Vietnamese vampire. Stefan Ziegler from WWF Germany emphasized that many of these species have thrived in the Mekong's unique habitats for thousands of years but are now facing extinction. Despite intensive agricultural and forestry practices over the last 50 years, the Mekong region remains critically important for its high biological diversity. Some of the newly identified species, including orchids and aquarium fish, are already present in trade, raising concerns about their future. New species are frequently discovered through field visits, collected and stored in natural history museums and botanical gardens for analysis. The urgent need for conservation efforts is highlighted by the ongoing threats to the greater Mekong region's rich biodiversity. Recent research from the University of Connecticut has unveiled a surprising twist in the study of senescent cells, revealing that some of these cells can actually play beneficial roles, particularly in wound healing. Published in the November 2024 issue of Nature Aging, the study emphasizes the complexity of senescent cells, arguing that a simplistic approach to eliminating them fails to account for their diverse functions. Researchers classified senescent cells into 2 distinct types based on gene expression, those expressing P16 and those expressing P21, each exhibiting different effects on the body. While senescent cells are known to accumulate with age and are linked to diseases such as diabetes, dementia, and cardiovascular conditions, they are also crucial for various biological processes, including tissue regeneration and immune system modulation. The study highlights that past research indicated the removal of senescent cells could extend lifespan, but this new evidence shows that not all senescent cells have negative effects. A specific study focusing on P21 cells revealed that their removal from young female mice accelerated wound healing by approximately 25%, indicating a beneficial role, in contrast to the effects of P16 cells. Future research is set to delve deeper into the role of P21 cells in wound healing, particularly in contexts like diabetes and aging, with the aim of developing targeted therapies. The findings suggest potential advancements in anti-aging therapies by focusing on eliminating harmful senescent cells while preserving those that are beneficial. Lead researcher Ming Xu stressed the importance of further studies into the heterogeneous nature of senescent cells and their varied functions across different contexts, underscoring the need for a nuanced understanding of these cells to enhance health and extend lifespan. The Lorandite experiment, or LOREX, is making waves in the field of solar research. Proposed in the 1980s, it stands as the only long-term geochemical solar neutrino experiment currently active, with the ambitious goal of measuring solar neutrino flux over approximately 4 million years. LOREX aims to investigate the stability of the Sun over millions of years by studying solar neutrino interactions with thallium, specifically focusing on measuring the solar neutrino cross-section. However, direct measurement of the neutrino cross-section on 205 TL has proven challenging. Instead, researchers have derived necessary nuclear physics quantities through bound-state beta decay rates. A significant breakthrough was achieved when the team successfully measured the half-life of fully ionized 205Tl81+ to be 291 days, a critical measurement for determining the solar neutrino capture cross-section. When neutrinos interact with thallium and lorandite ore, they convert it into lead isotope 205Pb, which has a long half-life of 17 million years, making it suitable for the project's timeline. With the data collected, LOREX can analyze the concentration of 205Pb in lorandite minerals, offering insights into the Sun's history and its influence on Earth's climate over millennia. Recent advancements by an international team at GS1/FAIR's experimental storage ring in Darmstadt, Germany, have provided essential data on the solar neutrino cross-section necessary for LOREX. Professors Gabriel Martinez-Pinedo and Thomas Neff emphasize the importance of nuclear astrophysics in addressing fundamental questions about the universe through this experiment. Dr. Ragandeep Singh Sidhu noted that this measurement could answer key questions regarding the Sun's evolution, marking a milestone in solar history research. The publication of these findings also acknowledges the contributions of late colleagues integral to the project's success, highlighting the collaborative nature of scientific research. A monumental glacial lake outburst flood has been documented for the first time in East Greenland, releasing over 3,000 billion liters of meltwater from Catalina Lake into Scoresby Sound. This event occurred between late September and early October 2024, marking a significant development in our understanding of glacial dynamics. The flood was triggered by 2 decades of meltwater accumulation that had been obstructed by the Edward Bailey Glacier. As a result, the glacier lifted and carved a 25-kilometer tunnel beneath it. The flood, which took place from September 23 to October 11, 2024, caused Catalina Lake's water level to drop dramatically by 154 meters. It released approximately 3.4 cubic kilometers of meltwater, ranking it among the top 3 largest documented glacial lake outburst floods. The energy unleashed from this event was equivalent to that of the world's largest nuclear power plant operating for 22 days, which suggests a potential energy generation of 50 megawatts, enough to power a small town. Despite the immense energy generated, challenges such as Greenland's remote location and lack of infrastructure complicate the feasibility of harnessing this energy. Fortunately, this particular outburst did not result in harm to anyone due to Greenland's low population density. However, it underscores a global risk, as approximately 15 million people are at risk from glacial floods. This event highlights the urgent need for further study of glacial floods. Climate change is expected to increase their frequency, necessitating the development of warning systems to prevent disasters in vulnerable areas. Researchers stress the importance of understanding these events better to issue timely warnings and prepare for future occurrences. Notably, real-time monitoring of the flood was achieved using satellite imagery by researchers from the University of Copenhagen's Niels Bohr Institute, marking a significant advancement in glacial lake outburst flood research. GLOFs occur when meltwater builds up and bursts through ice dams. Such events are becoming increasingly common due to rising global temperatures and accelerated ice sheet melting. In contrast, glacial floods in densely populated areas, such as the Himalayas, have already caused significant destruction and endangered millions, emphasizing the global implications of these natural disasters. A recent study from researchers at Harvard University presents a groundbreaking perspective on plant evolution, particularly in ferns. This research challenges the long-held belief that reproductive evolution in plants is a one-way street, suggesting instead that ferns can evolve backward to less specialized reproductive forms. This finding contradicts the traditional notion of irreversible specialization in evolutionary biology. Ferns exhibit a diverse range of reproductive strategies, including both monomorphism and dimorphism. Some fern species demonstrate the ability to revert from specialized reproductive forms back to simpler ones. This adaptability is partly attributed to the fact that ferns lack seeds and flowers, enabling them to adjust more readily compared to seed plants. The evolutionary process is complex and non-linear, often resembling a tangled web rather than a straightforward progression. This complexity is frequently oversimplified in educational materials, which tend to depict evolution as a linear journey. Such narratives can mislead, especially in the context of plant evolution. Dallow's law, which suggests that evolution does not revert to previous forms, faces scrutiny in light of these findings. Understanding the capacity of organisms to adapt or revert in their evolutionary paths is essential for predicting their responses to environmental changes. Rapid shifts in selection pressures can influence evolutionary pathways in unexpected ways, as observed with mammals following the extinction of non-avian dinosaurs. The misconception that evolution advances towards higher or better organisms is further illustrated by historical depictions, such as Rudolf Zallinger's 1965 portrayal of human evolution. Early vascular plants, known as telomeres, originally performed dual functions of photosynthesis and reproduction before evolving into more specialized forms over time. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.